Okay, so hi guys, I'm Katie. I'm a research fellow in NINDS, and we're going to do today a basic introduction and overview of resting state fMRI. So as a quick outline, we're going to start with some background and motivation, including some of the key findings in the literature. We'll go over some of the main approaches for analyzing the data. Then we'll talk about some issues surrounding both the methods and the interpretation, and finally discuss some of the emerging directions in the field. All right, so what you're probably familiar with already, this is task-based fMRI, and so the goal is basically to isolate some specific aspect of brain function by engaging people in different types of sensory or cognitive processes or different behaviors. And so if we were to look at the data, then we tend to mainly focus on the brain activity that's being evoked by this task. But now I want to show you the data from a slightly different view. So this is us playing it as a movie. So what you're seeing here is the data from all the voxels in the slice during the task. So it's showing us the fMRI signal changes going on at every voxel. And each frame of this movie is 1 TR of our fMRI acquisition. So this is going a little faster than in real time. And so if you're to look at the activity that's going on within that active motor region, you're able to see that it's increasing and decreasing along with the task. But you can also see that there's so much else going on all across the brain. And this is going on all the time, not only during the finger tapping, but also during the resting blocks. And so I think this really reminds us that, that the brain is, of course, always continuously active. And in fact, this type of ongoing or spontaneous activity accounts for most of the brain's energy consumption. So if we suddenly, show someone an intense stimulus, the extra energy expenditure you get there is just a small fraction above what's already a very large baseline. And so one question that we could ask is whether we can learn more about the brain by focusing on this spontaneous activity. And this is actually one of the main motivations behind resting state fMRI. So this is basically a resting state scan. So we eliminate the task altogether. We just scan people as they're resting and thinking whatever they happen to be thinking. Traditionally, we just instruct the subjects to just keep their eyes closed or maybe keep them open and look straight ahead, and to try not to think of anything in particular. Usually the scans are somewhere between 5 and 15 minutes long. And if we look at the data that this gives us, so for every voxel, we're going to get a time course of activity. And this is, of course, going to reflect whatever is going on in terms of their thoughts, their spontaneous neural activity, plus whatever noise happens to be in the data. And then the question is, how are we going to analyze it? How should we approach this? And it's not immediately apparent because, unlike in the task conditions, we don't have known conditions to compare in this data set. So we don't have anything going on specifically, so we don't have any sort of prior expectation about what this should look like, or what time points in the data would be interesting to compare to each other. We also have no idea what the subject is thinking during the scan, and we need to figure out what parts of these fluctuations are driven by neural activity and what parts are driven by noise, and so that turns out to be pretty challenging for resting state. It does turn out that there's quite a lot of information in this data, but that we need to think of other ways of trying to find that. So one of the major ideas in the resting state field is that instead of using the task as a reference point for looking at the time series, we're going to look at the relationship between the signals of different brain areas, and this is what's called functional connectivity. So in this little cartoon here, these 3 areas would be said to have high functional connectivity because they're expressing very similar activity. And so would these 2, and then maybe we would get that between those groups the dependencies are weaker. And so, so that's just the definition here. And from the perspective of systems neuroscience, one of the major reasons or motivations behind this type of analysis is that if by looking at which regions have similar time courses, then if areas are showing similar time courses, then maybe there's a direct or indirect interaction between them, that they're operating together in some sort of a network. But this is kind of complicated to actually interpret in practice because we're really not looking directly at connectivity itself. We're not looking at the physical connectivity, and we're not directly probing interactions. We're just really taking these types of time courses that we observe, and we're calculating a measure of similarity between them, such as correlation. So the next question you might ask is, are there any regions that actually do show consistent correlations with each other in the resting state? And now I want to show you this first paper by, in the resting state field by Bharat Biswal. This was back in 1995. And what he did was to first scan people with a task. So he did a motor task. This was a two-handed finger tapping task, and it gives you very nice bilateral activation in left and right motor cortex. And then he scanned people in resting state. So he made sure they weren't moving, so he recorded EMG. And what he found was that if you take some voxels, say in the left motor cortex, you take their time series, and you look at which other regions of the brain show the highest correlations with this time series, which ones have very similar fluctuations, he found that the areas that were most correlated fell within other parts of the motor network. So that's what this picture is showing here. So in other words, looking at the left and right motor cortex, these are very highly correlated with each other, and more so than in their correlations with other parts of the brain. And so this was an important early demonstration that areas that tend to operate together as known functional networks also tend to show correlations with one another in their resting state signals. And this was actually found not just for motor systems, but for also for sensory systems and cognitive systems. And so if you look at these panels marked rest, what's shown in each of these maps are areas that have highly correlated or coherent time series with one another. And you can see this direct comparison against regions that tend to co-activate with one another in tasks. So like here you get bilateral auditory cortices, you get voxels in the visual cortex, and even frontal and parietal areas that are thought to be involved in cognitive functions like working memory. And so if we think about this for a second, this has a pretty compelling implication, because it suggests that just by looking at this resting state data and just by looking at the correlations between regions, we're able to get all this information about which areas might be operating as functional networks. So we may not even need to use a bunch of tasks in order to find that. And then this would be really useful, of course, for studying populations where it's difficult or even impossible to have somebody do a task, like in small children or in patients who are unresponsive. So to talk about terminology for a second, these patterns we've been seeing are often referred to as resting state networks or intrinsic networks, and the areas within the networks are referred to as nodes. And it's very common to see pictures like this at about the spatial granularity, so about the size of blobs. And when we look at this level, we tend to find about 10 to 15-ish reliable patterns that we can identify across scans in individuals. And these are often given names based on the areas that tend to fall together in these networks, like you might see auditory network or visual network. But keep in mind that these names are definitely not standardized across the literature, so there's some patterns of areas that you might see referred to by several different names in different papers. One question that may be tempting to ask here is whether there's a certain number of networks in the brain. But actually this may not be a very well-defined question, because just like we can kind of design different tasks to probe, to try to get task activation maps with different levels of granularity, we can do a similar thing for resting state networks and try to study network organization at different levels of spatial and temporal detail. So we can think about taking any one of these subnetworks and breaking it up into more tightly connected subnetworks. So that's also something to keep in mind. These patterns can be identified really reliably. So here are scans from the same individual over many different weeks, I think. And you can see that these 3 different network patterns here tend to be very repeatable. And here's data from several different individuals for 2 scan sessions each, where you can again see that these patterns are very similar. We can also identify these over different brain states, like sleep, also in different species. I'm going to use the pointer instead because I can't find this. Like monkeys and rats, even when they're under anesthesia, and even in the infant brain. And the fact that we can actually see these over different brain states like this, like sleep and anesthesia, it suggests that these patterns are not just emerging by something like conscious thought or sensations, but also might be shaped like by for instance, the anatomic connectivity. So how does the physical architecture constrain this propagation of spontaneous activity? And perhaps other processes like autonomic activity or homeostasis. So I've been showing this pattern quite a bit, and now I wanted to spend a little more time describing it since it's been pretty important in the development of resting state. And so this is called the default mode network. And Marcus Rakel and others have found that these areas tend to show higher activation when people are in more passive resting state blocks compared to more focused or demanding conditions. And that's in contrast to many other regions of the brain. And so this is kind of odd and interesting network, and the fact that it showed higher activity at rest caused people to really think about what's going on in that resting baseline that we often use in our task studies. And so that helped to drive the study of resting state. So to recap a little bit, we find that, this is a resting state scan, again, a movie, that even in this super unconstrained and really complicated brain activity, there actually seems to be a high degree of organization when we look at things like the temporal correlation between areas. And these correlated patterns seem to align really well with functional networks. To highlight that correspondence a little further, this was a paper appearing in Science last year, and it shows the task versus rest correspondence at the level of the individual subject. So these three panels correspond to three different subjects, and this was their task activation map. Below here is the predicted task activation maps using only information in the resting state data after having trained regression models. And so pretty compelling that you can see that some of the spatial details are really captured just by the resting state data. So what I've been mainly focusing so far on is how stable the networks are, but actually there can be some degree of variability over different conditions. So for example, here is wake and deep sleep. So in deep sleep compared to wakefulness, you see a relative decoupling in the posterior and anterior parts of the default mode network. And it's also been really important that we are able to identify certain differences across neurological or psychiatric disorders. So here it was found that the hippocampus showed reduced functional connectivity in patients with Alzheimer's disease compared to age-matched controls. So perhaps this connectivity analysis can provide useful biomarkers. This is another example looking at whether levels of functional connectivity or correlation between PCC and medial prefrontal cortex tracked the severity of behavioral symptoms in schizophrenia. And so this variability may be quite useful. And just to give you a sense of the range of clinical applications of resting state, this is a table from a review article now several years old. And you can see that everything from schizophrenia, depression, aging, trauma, migraine, this is now being really, really widely studied for clinical applications. And there are now many more since 2012. So resting state has really taken off for studying brain networks and in searching for potential biomarkers. And some of the pluses is that we may be able to study networks without needing task compliance. And this also helps to make these brain networks accessible within a broader range of individuals who may not be able to do tasks. So next I'd like to move on to some methods, and I'm going to cover three of the primary approaches, and these are seed-based correlation, independent component analysis, and network analysis. So seed-based correlation, this is really straightforward and probably the most popular approach in the literature. And the idea is that we choose some kind of ROI or voxel, and we call it the seed. And then we extract its time series across the resting state scan. And then we take the correlation between its time series and that of every other voxel in the brain. And so we can show that on a map like this. So the value at each voxel here represents how strongly correlated it was with our seed. And then we might do some statistics and threshold that in order to get a statistical map. And so what you get here actually looks like an activation map, but the regressor in this case comes from the time series of our seed region rather than from a task regressor. And actually we can use the same general linear modeling framework as we do for task data analysis to do the seed-based correlation. In terms of some of the questions we might ask with this kind of approach, one might be, are there any areas whose correlation with your seed region are significantly different in two conditions? And another might be, are there any areas whose correlation with your seed is proportional to some kind of behavioral measure or outcome measure? So maybe something like treatment efficacy or learning and so forth. So if you do this for different seeds, these are some of the maps you might get. So this was a posterior cingulate cortex seed that's a major node of the default mode network. And if you take seeds within motor or visual areas, then you end up getting what we'd expect, which are motor or visual networks. We can also do this on a more fine-grained level with tinier seeds and high-resolution fMRI. And so here it looks like if you place a seed in different areas of visual cortex that corresponded to different eccentricity representations, so over here is an eccentricity map for reference, then it looks like the highest correlations in each case are following those areas that have similar eccentricity and in both of the hemispheres. And so even when people are resting with their eyes closed, you can still see by looking at correlations that that it seems to reflect this functional organization. So AFNI has made a nice tool for correlation analysis. This is called Instacore. And the way it works is basically you click on a voxel, and what you get are instantly the correlations with that voxel across the brain. They also have a group level version, so that basically does this, does a group analysis for this. And this can be a really helpful way of exploring your data, though of course you have to avoid just clicking around and fishing for significant effects. And another thing that this can do is give you a nice way of quality checking your data. So sometimes artifacts can pop out really obviously in correlation maps. And as an example, these are two different data sets. And in this one, if you click on this voxel here, you get this pretty normal looking correlation map. But over here, you get this crazy pattern, and this turned out to be some kind of hardware artifact. So one of the considerations for seed-based analysis is that we obviously have to choose where we're going to put our seed and what extent we're going to use. So how many local voxels are we going to average across to get a time series, a seed time series. And so some ideas include using a region from an atlas. You could use a published coordinate from the literature. You might also manually draw it on someone's high-resolution anatomic image. Or have somebody do a task and look at their activation map in order to find the specific voxels in the brain, maybe corresponding to something like the motor cortex. And one thing to be aware of is that the results can actually depend pretty strongly on where you're going to place your seed. And so this is an illustration of what happens if you move a region, a seed region, across this little line here. So this is a more blown up view. And so as you move your seed, you're going to trace out a series of different correlation maps. And so if you move it a little too far, you can actually end up in a different functional region and get a really different correlation map. But you might also recognize that there's something pretty interesting about this, which is that this might help us figure out where functional boundaries are in the brain. So maybe we don't know where this red region transitions to this blue region, but if we see a really rapid change in the correlation pattern along the way, then maybe that means that we're jumping into a distinct area. And that idea has been proposed in this paper, and it's been used by a number of others as well to try to find functional parcellations. And this is a visualization of that idea as a movie. So here they're moving this seed around the parietal cortex. And as they move it, you can see how the long-range correlation patterns are morphing around as well. I'll let you watch it for a second because it's really cool. Okay, so the second method we're going to cover is ICA, and this is also a really popular method. And so Here I've just shown you basically the inputs and outputs. So basically what goes in is your data. So these are the sequence of bold volumes at each TR for the scan. And what you obtain is a set of maps or components, each of which we can think about as a network. So when one uses ICA, the words component and network get used interchangeably. And I didn't draw them here, but each of these time courses, each of these spatial maps also come with a time course that reflects essentially how active it is over time. And in contrast with seed-based correlation, this is a multivariate approach, and that essentially decomposes your data into a set of networks all at once. And so it's data-driven. You don't need to specify some seed region beforehand, but you do need to specify some other parameters like the number of components you might like it to return, although there are some methods for doing this automatic dimensionality estimation. And you'll get a number of components that look a lot like realistic networks, and you'll also get some that look like artifacts in the data, and that makes ICA a useful tool for analysis as well as trying to reduce noise in the data. So if we just look a little more closely at how it works, the basic idea is that given any time frame in your data, this volume can be modeled as a linear mixture of activity coming from these different networks. So at time 1, we have this A1 times component 1, A2 times component 2, and so forth. And note that these weights can change across time, but in what's called spatial ICA, the networks are what are remaining. The spatial maps are what are remaining fixed. And actually, if we take one component and look at its series of weights across the scan, that's giving us this useful time course of how active that network is at a given time. Oops. So when we just run ICA, we only start with our data, and we don't know anything about the weights or the components. And so we have to estimate those, and ICA does this by placing some assumptions on the data, like that these spatial maps, or these maps are going to be spatially independent. And so it's basically trying to blindly unmix these from the data. But something that's quite cool about this is that even though we're using this kind of blind mathematical criterion, it returns these really biologically plausible networks, just like we get with seed-based methods. And so these are some components that result from running ICA over a group of healthy subjects, so you can see the usual networks. And even though there's not a really clear mathematical relationship between ICA and seed-based correlation, they both tend to hone in on areas that have highly coherent activity, and so that's one reason we see them in with both methods. One consideration here is that the appearance of the components may be quite different depending on the number of components that you're requesting. So if you say you want to divide your data into 120 components or some very large number, then you're going to get sparser and sparser maps compared to asking for really low levels or only 30 or something. And so you might even start to see some of these familiar networks split apart into different components. There's an extension of ICA known as dual regression, and that can be helpful for getting around this in some ways, because the idea is that if you start with some a priori spatial pattern, then you can try to estimate what the weighting of that pattern is in an individual data set, and that makes it easier to compare corresponding things. So I won't describe that in detail, but here's the reference. So as I just mentioned, another nice property of ICA is that it kind of contains this built-in way of denoising the data. And that's because in addition to seeing some kind of normal neural-looking networks, it also conveniently splits off a number of artifacts into several separate components. And so by definition, that means that these remaining networks contain less of that artifact. And so one idea is to carry these forward in our analysis. But another might be to just try to eliminate these noisy components and then just reconstruct another four-dimensional data set so we can do other things besides ICA on it. But as we're going to get into in a second, this requires us needing to somehow objectively choose which components are noise and which ones are not noise. And so we need to develop some ways of doing that. But before I leave this, I just want to say that this is really a nice way of kind of discovering hidden structure in your data. So maybe you can see new patterns or weird noise patterns also that you may not have expected to be there. So in terms of identifying noise components, one approach that's been suggested is to use machine learning to try to automatically classify components as noise or non-noise based on your training data set. And that's what's identified in this FIX program, which is part of FSL. And another approach that was developed in Peter Vandettini's group here is based on multi-echo ICA. And this basically uses the property that bold signals, like you don't expect in real brain networks, that these scale as a function of echo time, but non-bold signals will not scale in that way. And so if you, instead of collecting just an image at a single echo time per TR, if you collect multiple echoes and you run ICA, then you may be able to group those components, or basically rank those components on a spectrum of how much it does or doesn't scale with TE. So whether it's likely to be a real component or whether it's likely to be an artifact, which can be really helpful in trying to separate those. And most kinds of artifacts tend to separate very well into the low end of the spectrum, but you do have some trickier ones that actually arise from bold mechanisms, and they have a large spatial overlap with real-looking networks, and so these are not as easily separated with ICA-based methods. And this slide just summarizes a bunch of things that we talked about for your reference. I also wanted to point you to this review article, and it covers some of the trade-offs between ICA and C-based analysis, as well as some other considerations. So the third class of techniques is called complex network analysis, or also graph theory analysis. And the building blocks of this approach are basically nodes and edges. And so we're going to represent our fMRI data as a set of nodes connected by edges. And the nodes are often taken to be distinct brain areas. And edges, the strength of an edge between two nodes represents some measure of association between them. And this is commonly something like how correlated their time courses are. So once we've specified these nodes and edges, then we can try to characterize properties of the overall network organization, such as finding groups of nodes that may be highly interconnected within each other and maybe not so much between each other. These are referred to as modules or communities, just like communities in a social network. And we can also focus on properties of individual nodes, like how much a node, or how many connections a node has, or how much it tends to bridge between two different communities. And so for the brain, maybe that means something like integrating information across kind of disparate areas. And there are many possible measures that you can use for this kind of thing, and these are just a few useful references. Generally, the way that we define our nodes and edges at the beginning is really critical, because it's going to affect all the subsequent results that we're going to get. And we're going to, we want to basically carve up or parcellate areas into meaningful units rather than just arbitrary boundaries. So if we happen to carve out arbitrary boundaries without regard to the underlying information, then at the end of the day, we might end up with results that are either really hard to interpret or not what we intended to do. But ways of best parcellating the brain into homogeneous regions, this is actually a tricky problem, and there's a growing area of research focused on how we might be able to do that. Okay, so now let's get into some of the issues, and I like to quote this sentence from a review article, which is that functional connectivity is a powerful but ambiguous mapping tool, and I think we can actually describe resting state in general as being powerful but ambiguous, because even though we can detect all of these interesting things in the data, it's usually not clear how to interpret all of these things that we're finding. Now, one reason is because we don't know exactly what are the underlying processes in our time series. So there are many different sources of noise as well as neural activity that are embedded in them, and these are not so easy to isolate or measure. And we're dealing with noisy data, and the results are often sensitive to both our modeling and our pre-processing decisions. And we also have a limited understanding of how the signals and the correlations we observe in resting state data relate to anatomic connectivity as well as electrophysiological signals, and this is also an active research area. So one type of artifact that makes resting state tricky comes from physiological processes like natural changes in how quickly or deeply we're breathing over time. And the problem with that is that it causes common bold signal modulation across many areas of the brain. And so this can cause spurious correlations, because if you imagine placing a seed in an area that contains a lot of breathing-related effects, then you're going to get inflated correlations with other areas of the brain that also show breathing effects. And then, if you have systematic differences in breathing across subjects or across groups, then you might end up finding as well systematic differences in correlation levels. So it's a good idea to try to minimize any known sources of artifact as best as we can. And for physiology, I always recommend, whenever possible, trying to record the breathing or the cardiac activity during the scan, and that's usually readily available with most scanners. Head motion is another common problem, and what Jonathan Power pointed out in this study was that if children are having more head motion than adults, then we can get what looks like real age-related changes in correlation between brain regions, but what it may be really picking out, what may be really happening is that we're getting age-related changes in the amount of head motion. In resting state data, telling signal apart from noise is pretty hard, because if we had a task going on, then that would be somewhat helpful, because we would at least have some model of our expected activity, or we could do something like average across trials in order to improve our SNR, or our signal-to-noise ratio. And another point is that instead of comparing our data to a noiseless task activation model, we're usually comparing the time series of one noisy brain area to another noisy brain area. And so if this isn't carefully treated, then we could ultimately draw the wrong conclusions about our networks and connectivity. It's also really tricky to evaluate whether noise reduction is doing the right thing in your data, because we don't have ground truth, right? So, several years ago, there was this famous debate over whether the default mode network and the attentional networks are negatively correlated in resting state. So some early papers suggested that these networks did have this anti-correlation relationship, which is interesting because these two systems are thought to reflect internal or externally oriented processing and maybe kind of keeping each other in balance. But these studies had used a pre-processing step known as global signal regression, and that's basically you take the whole brain average time series and you regress that out of every voxel in the brain. And so it's a pretty aggressive way of trying to remove anything that's common across our voxels. But incidentally, this can mathematically introduce anti-correlations in your data. And other people had some trouble replicating the anti-correlation without using global signal regression. But what's the right answer here? Because, in this case, global signal regression may be just too aggressive and is introducing negative correlations artificially. And in this case, we might not be removing enough common noise in order to see those anti-correlations. And that's why these debates can go on for a long time. I'd say, it's the best thing, if possible, would be to use as many kind of unbiased or model-driven methods for removing the noise as we can, like actually recording physiology and trying to project out time series relating to these recorded physiological waveforms, or using something along the lines of multi-echo ICA that gives you a little bit more objective way of separating noise from non-noise. So here I think that the current consensus is that there are anti-correlations in the data even without global signal regression, but that they're not quite as spatially extensive as when you have global signal regression, and they may also to some extent be dependent on a subject's brain state. So now let's talk about some of the neural processes and states that may underlie resting state signals, and one of the things that may come to mind immediately is mind wandering, or just thinking about various things. I mean, that's just what we do. So several studies have shown that the correlations between brain areas can differ depending on the types of thoughts people might be having during their resting state scan. So here they actually gave people questionnaires after the scan and tried to figure out were they thinking more visual thoughts or more language-like thoughts, and then of course they could find differences in the network structure. And here, I've forgotten the labels, but they also found that if you're kind of singing a song in your head versus counting backwards, then you're going to also get reliably different patterns. Another thing that happens is that we get drowsy and we even just fall asleep in the scanner, and especially in resting state because it is really boring. And unless you're studying these brain arousal or sleepiness processes for your research, this is going to be a confound in your studies. And so some of the things that happens when you become drowsy are, it seems like the signal amplitude or the fluctuations tend to become larger. And another thing is that there seem to be more widespread positive correlations in your data. So this was a data set before and after they gave subjects caffeine. And in order to tell if someone's getting sleepy, that's kind of tricky unless we record other things in our scanner, like EEG or watch the subject's eye behavior and so forth. Sometimes these measures are not available, and so we and others have been working on some ways of trying to infer vigilance levels across the scan from the fMRI data itself. Now I want to talk a little more about the link between functional connectivity and structural connectivity. So there are many examples actually showing a really tight link between the two. Here they found that these small subregions in the cerebellum show the strongest functional connectivity to areas of the cortex to which they're known to anatomically project. And similarly in the thalamus, different subregions also show preferential correlation with the expected cortical targets. And so this is really amazing correspondence. One of the ways we can more directly probe that correspondence is with special populations. So some people are actually born without a corpus callosum, this major white matter band between the hemispheres. And here they find that even in a task, they were able to get both areas, both hemispheres activating, but when they looked at the interhemispheric correlations in the resting state data, that was much weaker in the resting state. And they had no corpus callosum. So this is seems to be, structural connectivity seems to be a critical determinant of functional connectivity. But over here, they looked at correlation maps before and after a subject had surgery that removed the corpus callosum. And so this column is before and this column is after. You can see that for most networks, this bilateral, the typical bilateral patterns disappear. But there were still some regions that had some correlation between both sides, and it was suggested that this is perhaps because correlated activity can be conveyed through indirect structural connections. So there's not a one-to-one relationship between structural and functional connectivity because we can still have these indirect pathways. Another situation, I didn't draw this here, but is that if you have, there's going to be cases where you have two areas that look correlated because they're both correlated with a third common source that's maybe driving them. And so that makes it hard to say whether those regions A and B are actually talking directly to each other just because they're correlated, because we might have this common driving node. So finally, how does resting state signals, how do these relate to more direct measures of neural activity, like from electrophysiology? Well, one of the robust findings is that there seems to be a correlation between spontaneous fMRI signals and local field potentials, particularly the power in the gamma band. This is similar to what's been shown for task-evoked activity. And accordingly, it's also been found that the long-range correlations between gamma LFP that are measured at different sites, in this case this is different ECOG electrodes, that these show correlation patterns that resemble the fMRI resting state networks. This has also been demonstrated for other features of the electrophysiological signal, like this slowly fluctuating activity, the slow cortical potential. And there have been many studies now on other frequency bands, like alpha, beta, theta. And there the picture seems to be somewhat complicated. It may even be that multiple frequency bands kind of are correlated with the same resting state network, it's still not really clear whether there's like a kind of a uniform relationship between these. So overall, it seems that we can say that there is correspondence between resting state fMRI and electrophysiology, but the way that all of the different features specifically relate to each other is still being investigated. Lastly, I just wanted to touch on a few of the emerging research directions. And one of them, I think, is combining resting state with brain stimulation technologies like TMS or TDCS. And one current direction is basically leveraging functional connectivity to try to define targets for non-invasive stimulation therapy. So say you want to stimulate a deep brain region, but using non-invasive methods, you can only access more superficial areas, like in the cortex. Can we map out the functional connectivity between these deeper areas and more cortical areas to find a cortical site that we might target in order to reach that deeper area? And so that's what they did here, and they used TMS. We can also use brain stimulation to study neural mechanisms, like things like the causal relationships between brain networks. And so here we excited or inhibited different nodes of various resting state networks and looked at whether perturbing one node could lead to a change in either the correlations or the frequency properties of other networks. Another direction that's growing is the so-called dynamic connectivity. So usually in the work I've described, we take the entire length of our scan in order to compute some metric. But the idea here is, what if we start breaking that up into smaller time windows and looking at how the associations between different brain regions are changing over time, and perhaps these more dynamic features might carry some important information about cognitive states or brain disorders that would be missed if we're just averaging over all this to create one picture of what we think our associations are. And so there's been a lot of work in the methods development and applications of this area, and it's actually going to be covered in a whole other lecture later on this summer. Another idea is fingerprinting, or looking at whether features of fMRI data can capture individual differences. So Emily Finn finds that these correlation matrices in resting state data as well as task data, using the functional connectome or the human connectome project data, that these could distinguish reliably between individual subjects, and also was very predictive of cognitive measures like fluid intelligence. And in addition to the Human Connectome project, there are really a lot of public data sharing efforts for resting state. And these, as these massive amounts of data are available, it's allowing us to find effects that we really wouldn't see in smaller data sets. And there are also data sets for clinical populations as well, like autism or Alzheimer's disease. So to summarize, resting state data have this really rich organization, and it's being really widely used for clinical applications in basic neuroscience. There are a lot of methods out there for analyzing the data. I mean, we really just barely scratched the surface of all of the approaches that are being used now in resting state data. And I think it's really important to be aware of how these methods work when you're applying them to your data and their trade-offs. And I'd also recommend staying as close to your data as possible. So look at the raw images and the time series a lot to understand what's going on, because things like correlation can be a really kind of indirect measure of what's actually going on in your time series. And so it can be helpful to look at all of these things whenever possible. And finally, there's a lot of sources of variability, both from neural sources and noise. And so that can make things a little bit hard to interpret. And of course, we can't always control for these things, but it's important to be aware of what might be contributing to your results, and just to correct for things wherever you can. So that's all. Thanks a lot. Are there any questions?
You mentioned that there are different ways of measuring how well collected two regions are. correlation is one of them. Could you talk about some of the others?
Yeah, so the question, since they're recording this, I have to repeat, is besides correlation, are there other metrics for looking at the similarity between time series? Well, one of them is mutual information, so that takes into account kind of higher order statistical similarities between areas. People have also tried looking at directed measures, so is there a kind of a time-lagged relationship between region A and region B? One of these approaches is called range of causality. Though in fMRI, we have to be really careful about these kinds of things because we're looking at this blood flow related signal, and sometimes there are blood flow delays across the brain that are not correlated, with the neural delays, and so we could kind of sometimes get the wrong answer there. Yeah, and I didn't really cover it, but there's also methods where you take a rather small set of nodes, or actually can do this for larger numbers of nodes as well, like structural equation modeling or some kind of effective connectivity models where you're trying to fit these kinds of directed or undirected relationships between a set of regions. Yeah. How can we know the neural connectivity instead of the
connecting from
yeah.
From the blood supply?
So I'm not sure I understand what you mean. Sorry.
This connectivity is caused maybe caused from the blood supply.
Oh, okay. So is connectivity caused from the blood supplies? Yeah. So actually, one of the initial big doubts about resting state is whether this is all blood flow effects, and actually this vascular territories are also nicely bilateral and things like that. So maybe we're just getting these correlations based on blood supply or other things like. There's been really interesting work showing relationship between kind of low frequency cardiac oscillations and brain activity. So it's we can definitely get correlations between areas based on things like blood flow. I think there's enough evidence using methods like ECOG or other more direct recordings invasively, also in animals, that shows that it's not fully driven by those things. But I think as a field, people are still working on how to best correct for these kind of non-neural influences like blood flow or like changes in the breathing rate that can also introduce a whole bunch of coupling but not be related to the local neural activity. Okay, thanks a lot.
Good job. I really enjoyed it. You updated it.
I did. I always try to a little bit, but I had less time than usual. I got sick this weekend, and I was like, oh no, this is the universe telling me not to update it too much. And I was like, am I going to be able to talk? I was like, yeah. I saw you're doing one this year.
Yeah, I know. Well, I actually wanted to see how
this thing works.
Yeah, I mean, how do they record? I think they're supposed to record it. So
yeah. So Dorian will come and he will set up Camtasia. And he did that for me, which was really nice.
Oh, okay, so I can't use my own laptop. I have to use.
Unfortunately, no. So yeah, they
laptop,
they
okay.
use but
and it's a Mac, but it has Mac PowerPoint.
Yes. So yeah, if you wanted to check it earlier, I'm sure Dorian and them could.
I think it'll be fine. I'll just
yeah.
Movies or something like that.
Yeah.
Do you have a question?
Yeah. Kind of updates for you too.
Oh, okay.
Hi, my name is Rachel. I just started as an IRTA with NINDS in the clinical epilepsy section. So we use, and Dr. Peter. Yeah. So